Hello, everybody. Welcome to the Labroots webinar. Today, we are going to talk about from the data to biological insight using QIAGEN OmicSoft and QIAGEN IPA, and specifically for the single-cell sequencing of a normal human liver. So the objective to explore the cellular landscape of the human liver in that particular case, but it could be true for any type of tissue or organs, is to understand what cells comprises a normal human liver. So how many distinct cell types are in a representative liver sample? What gene expression markers best distinguish cell type within the liver? Do published cell type signature correspond to the liver cell clustered by expression similarities? We can also try to understand what is the biology shared between these clusters, what molecular pathways and biological processes are highlighted in the clusters, what transcriptional programs underpin each cell type. And also, with our technology, we can try to discover hidden biology by comparing that specific single-cell analysis of all the different clusters to a set of over 60,000 curated analyses. So the agenda today will be to describe for you what is the QIAGEN meaning of sample to insight. We're going, second part, to talk about the QIAGEN OmicSoft Land. How do we curate the single-cell data set? Then we are going to explore from the OmicSoft Land to Array Studio, which is the software that do the data analysis, where the data is going to be processed. Then from Array Studio to QIAGEN IPA, we are going to send the data to discover and highlight the biology of the different cell types in the liver. Then we will try to explore hidden biology, and I will explain what it means. Then we will bring some conclusion. The QIAGEN sample to insight, it's a very important notion that equip any researcher and scientist in the lab from the sample collection and stabilizations to the interpretations. And today we are going to talk about three specific software that we have developed over the years. QIAGEN OmicSoft Array Suite for the data analysis, and QIAGEN IPA, or QIAGEN Ingenuity Pathway Analysis, and QIAGEN OmicSoft Land Explorer for the biological interpretation. The QIAGEN knowledge base indeed powers the IPA, Ingenuity Pathway Analysis. And we do that because we have accumulated literature findings over the last 20 years. And all these findings that are manually curated or using expert assist findings, we have structured them in a knowledge base, model knowledge base. So every content and every findings, as we describe that, are well structured in a way that the software, IPA, is able to retrieve, based on your expression data, very key and important findings that is pertinent to that data set. The content of IPA is based on what we call a finding. This is a typical finding. And what's key to remember here is the content is contextual and have direction of effect. In other words, we capture every important features of the findings in order to tell us on the right that a particular gene, here, NFE2L2, downregulated in a particular experiment, when it's connected to that GSTM3, and we know the connection, we know the interaction between these two genes, we can predict the effect of that downregulation on NFE2 on that gene, in that case, predicted to be inhibited. And all of that is due to the context and all the important information. Direction of effect on the gene expression, mutation type, if it's an upstream or downstream gene, what type of tissue, what type of species, and so on. So all these 7.1 million findings help us to understand what's going on and to predict the pattern of activity. particular case we have developed today, it's come from a public data set put on the NCBI Geo, and it's about a paper that has been published in Nature Communication in 2018 by McParland and its group. And the title was Single-Cell RNA Sequencing of Human Liver Reveals Distinct Intrahepatic Macrophage Populations. So they have proceeded to a single-cell RNA-seq of whole human liver cellular landscape from liver graft of 5 healthy neurological disease donors. They have a particular procedure. They dissociate the liver tissues in order to not kill the cells per se. And they have followed a single-cell sequencing protocol using 10x genomics single-cell 3' V2. Then they sequence on the high-seq Illumina 2500 using Cell Ranger for the pipeline. In our case, we have used the alignment to human B37 using our proprietary aligner, OmicSoft aligner, and we have quantified using RLCM and proceed to obtain dimension reduction using t-SNE. So the data which is processed in OmicSoft is going to be sent directly through IPA via a simple plugin. It's very easy for biologists like me. You are going to process the data, you send your expression file, and in IPA, we are going to interpret the biological data set. And in return, we are able to compare your own analysis or analysis to a set of 60,000. I will explain that in later slide. So OmicSoft Land processing pipeline starts with retrieval of public submitted projects where relevant to a specific disease have been submitted to a variety of public resources, including NCBI, SRA, GEO, RS Express, ENEA, and a subset of the Human Cell Atlas. All of that have been curated in a single cell land pipeline. The key to the OmicSoft curation is, in fact, the collaborative process between the biological curator and the bioinformaticians. And you can see here there's multiple steps between these two pipelines. The interaction is essential. The curator will perform a number of metadata extraction and curation steps, including automated metadata extraction from submitted geo projects. And in addition, single-cell specific metadata fields will be extracted as well from the project and from the paper. That will be followed by the curation of the project and the paper to apply a very controlled vocabulary columns that can identify key statistical comparison and identify as well expected number of cell types. The data processing team downloads the raw data from the FASTQ file using the advice from the biologist curator. It defines the pipelines to analyze these data depending on if you have a UMI or non-UMI type of data. I will explain that in the next slide. Then the full data processing pipeline resulting in quantification of the genes will be obtained. Using the advice of the biological curator, identifying the number of different clusters expected in a t-SNE analysis, the data processor will do an initial analysis and dimension reduction of the sample to identify it and to generate a t-SNE plot. That will allow us to identify initial number of proposed cell cluster or cell types. These data will be shared back with the curator to review and identify the proposed cell types and how these compares to the original paper. If there is any discrepancy, then the additional advice will be sent to the data processor to rerun the t-SNE and identify the proper number of clusters to best represent what has been founded in the original paper. The data processor will run a differential expression analysis using SERAT. to identify the top differentially regulated genes between the different clusters, and these cell-specific signatures can be used to identify cell types and different express genes as well. These data are entered into the Land repository. And this is an example of some of the source for the dataset processing and metadata curation the biologist relies on. It includes gene expression omnibus submissions and sometimes the data from the methods of the papers and the result papers. You can see here there is a variety of different single-cell library preparation technology that all requires different methods for the pre-processing and the analysis of the data. For example, you have UMI, unique molecular identifier, based technology that will use the 3' tagging of unique molecule to minimize the amplification bias artifacts. And then there is also non-UMI based technology that tend to measure a smaller number of cells but have a higher coverage, and they don't have the 3' bias. So the manual curations of the single data from a domain expert is key, right? And these are some of the resources that are used by the OmicSoft curators to analyze single-cell data. The source paper, the gene expression, which you have that on the submissions, which has many columns that can be extracted to the project level, the sample level metadata that are submitted for each individual biological samples, the actual sample data files, which are compiled into a metadata framework and controlled for certain key terms such as cell type sources. These sources are compiled into a metadata matrix to allow you to find a sample that are of interest for you. In addition to the geo-based submission, additional metadata are extracted from the paper itself to help advise the curation of the metadata and to advise the bioinformatician in their analysis of the data. There are automatically extracted metadata columns that are then formatted to meet a mix of curation standards using controlled vocabularies and formatting with additional information extracted from the publication. There is additional manual curation based on the disease therapeutic area and more focused disease, each individual subject, disease state, and the tissue from which the data came. In addition, as available, any sort of recommended metadata such as cell types, identities, and treatment status, and so forth, are also applied to the individual samples and are all applied with a controlled vocabulary. In addition to the automatic and manually extracted key metadata, there is also additional project-specific metadata that can also be included into each of these single-cell projects. Animal strain, land-specific, disease-specific information, depending on the specific therapeutic area that could be analyzed, and any platform-specific information that would be relevant to the biologist and interpreting these, such as the library preparation and the presence, for instance, of ERCC spike-ins for normalization. Again, what is key here is the interactivity and iterative curation between the biologist and the bioinformatician to have the best possible single-cell line project, the most reliable, the most faithful to the original paper. So this is particularly important for the generation of the dimension reduction plots, such as the t-SNE that you can see on the right. Based on the source paper, the biologist advised the bioinformatician about which individual samples should be included in a particular t-SNE analysis, as well as the expected number of clusters, which approximates the number of cell types expected to be found in this particular analysis. The bioinformatician then runs an initial t-SNE analysis, including k-means clustering to identify the predicted cell cluster. These data are returned to the biologist to review the t-SNE result, then adjust the number of cluster settings to match the published result as best as possible to make sure that samples or cells are not being divided arbitrarily. Then the bioinformatician runs the t-SNE pipeline to match the publication. An additional curation may be added by the biologist to help identify what the individual cell types are, using an annotation of the paper of the individual gene signature for cell types and domain knowledge of the biologist. And then the final project is submitted for publication into the Land. So within the Land framework that you see here, the way you would find a new project of interest is simply to browse the available projects showing here. On this bar plot, you can see that in this case, we've broken down the different projects based on the main tissue of origin. We're looking at liver here for our cases. We have also project metadata that are of interest. When you find a project of interest, you can actually click download that project into your server project, and the full package will be in your project folder. That's what we have done in that case. So we have uploaded that specific project, the GSC 115469. It's a UMI project. It's about the liver, single cell of a normal human liver, sorry. And what is put into, download into our server project is all these files, free computed files. For biologists like me, it's very easy and it's very quick. So you get the cell curation type, you get the quantification table, you get the differential expression in the inference report, you get the cell clustering pattern. You are going to discover the t-SNE. You have also the top gene markers using SERAT here. And you have a set of curated project information, such as I have described, the cluster, project, and sample metadata. That allows you, based on these heavily curated projects, to simplify, and this is what I've done in my case, to some biological cluster of interest. So based on the k-means 20 that have been presented in that papers, I have, for the sake of that webinar, simplified to a set of 12 clusters with some namings: alpha beta T cells, cholangiocyte, erythroid, hepatocyte, and so forth. In that screenshot, you are going to see that we use SERAT to obtain the gene markers. And we can also, using same SERAT, to detect the highly variable gene between the clusters. So from that step here, from which you have downloaded from the single cell Land project, to your own customized analysis, if you wish, it's extremely simple because you rely on the cluster cell type that has been predefined previously. So this is what the first t-SNE, based on the k-means 20, was obtained after curating that single cell analysis of a normal human liver. And that matched exactly what the paper has found: multiple clusters for hepatocytes, for endothelial cells. And from that, you can actually obtain SERAT test, which is equivalent to an inference report, basically a differential expression profile in which you can compare one versus the other clusters, or 2 versus the other cluster, or 19 versus the other clusters. And for each of them, you will have a for change value, a log2 transform, a p-value, and FDR values. That report is extremely easy to get, and you send that to IPA. But as I said, because the curation has been so well done, I can even go further and to define my own biological clusters. And here, this is a representation of the t-SNE based on the cluster cell type. And that match pretty much what the paper is saying. For the argument today, I have simplified the 5 clusters found in hepatocyte as one, the alpha beta cells, and many others that are described here. So 2 sets of type of cells in the liver. There's the parenchymal, which are the hepatocytes per se, and the non-parenchymal, which are basically the immune cells that has been discovered in that paper. Same as previously, I could create an inference report, basically an expression profile using SERA to define highly variable gene and top gene markers, and I would like to compare each of the previous biological cluster that I found. For each of them, I could get a comparison, hepatocyte versus others. I can set some features of interest, minimum observation per gene, 3, minimum of genes per cell, 200, and so on. Doing that, you obtain these in the inference folder, this type of comparison. Hepatocyte versus others, hepatic stellate versus others, and many others. So, this is exactly what I have proceeded. I wanted to look and understand when you compare one cell type versus the others, what are we going to obtain in terms of biology? And that should tell us a lot about the liver. This is what we have done. So we have set all these different 12 clusters and compared to the set of others, hepatocyte versus the rest, plasma B cell versus the rest, and so on. For each of these comparisons, we can get a volcano plot. You can tell here, on the right, the upregulated gene, in that case, in the hepatocytes, and on the left, which are supposed to be green, the downregulated genes in the hepatocyte versus others. That report can be sent directly to IPA. But before doing that, we have another feature which is really cool. We are going to use the scatter color view to visualize gene expression across the cluster themselves. To do that, we're going to use the design table, which has been provided when you download the project. You are going to select the table data, the cell map T1 score. You are going to extract the V1, V2 coordinates and add that to the design table. Here, this is the cells, tissue, disease state, and the V1, V2 coordinate. Because of that, I'm able to now visualize the expression of one particular gene in that t-SNE. And this is the case here. I selected the TNF-F12, which is a cytokine ligand for TWIC-R. And that is expressed in the liver, as you can see. But specifically, it's expressed in the lymphocytes, apparently, in some endothelial cells, and some macrophages. The group of hepatocytes, it's a little bit weak. The expression is weaker. But we have another tool as well. When you have QIAGEN IPA, and you obtain a gene view, and you select that particular gene, you can ask the questions, what is the expression of that gene in different tissues? To do that, to obtain that image here, we use a link that sent us the information to Land Explorer. Land Explorer is a web-based version of OmicSoft Land. It's very easy to use. And in one click in that case, I was able to look in blueprint B38, which is a type of land, on specifically normal immune cells where TNF-F12 is expressed. And you can tell in one click that gene is expressed big time in peripheral blood, and you see other tissues, much less in the liver. This is a log2 transform. So that information from the scatter color views, you can actually expand that and understand what is the expression of that genes in other tissue using the link from IPA to Land Explorer. It's very easy to do. SERAT, I told you, identifies top hepatocyte marker genes. And here I selected 9 of them, and this is the group of hepatocytes which is highlighted here, the top right. Many of you will recognize these genes, HP, VTN, and many others. You can do that as well for the Kupffer cells for the example. These are the 4 markers that has been identified in the paper. Clearly, we match exactly what the paper is saying. That is using the SERAT. You can also identify highly variable genes between the clusters. In fact, we like to identify genes that are highly expressed in some cell type and lowerly expressed in other cell type. And I'm going just to give you 2 examples today, STAT-MIN1 and CD52. And here this is the representations using SERAT as well of the expression, variation of expression of that particular STAT-MIN or CD52 in the different clusters that we have identified. So you can see there is a high variability. That can be used for further understanding what's going on in that liver. I told you the data is being processed in OmicSoft, the project sent to RS Suite, which is the data analysis processor of OmicSoft. And now you can send the data directly to IPA. And IPA is the power tool that helps you understand what is the biology in your data set. So we are using an API that basically sends the information of the data set to IPA, in which you can eventually set even the analysis, describe what's going on, set your cutoff, and send that to IPA. So once you are in IPA, the analysis is done. Here, this is hepatocyte versus others. We set up some fold change, some p-value. This is just a screenshot of the summary of the core analysis, what we call a core analysis. And basically, we give you the top canonical pathways, the top upfront regulator, and I will define for you what it is later, and many other informations. So let's go to the core of the data itself. So this is a single-cell RNA-seq sequencing of normal human livers, and it does reveal cell-specific canonical pathways. Here I have established a comparison using the comparison analysis in IPA. We are using the canonical pathways features, and we compare them in all these different clusters. Hepatocytes are in that first column, and all the other clusters are here. On the left, I've looked at the metabolic pathway %, and on the right, I've looked at the signaling pathways. Just looking at that comparison, it gives you an idea of what is the role of the liver. There's effectively a liver specificity in metabolism function. A lot of the functions are predicted to be highlighted. Orange will give you the prediction pattern of activity, blue of inhibitions. So in the hepatocytes, you see a lot of the canonical pathways here that I'm going to show you in the next slide that are predicted to be activated. And some of the signaling pathways are predicted to be inhibited. Inversely, a lot of the metabolic pathways are not activated. They are inhibited in many of the immune cells that have been discovered in that single-cell sequencing. But they are activated for signaling pathways in that cases. So a lot of pathways related to T cells, B cells, dendritic cells, NK cells are effectively predicted to be activated. So these help you to understand the immunobiology and the metabolic functions of the liver per se. Just one comparison can give you that. But let me explore a little bit. I show you here 4 canonical pathways that are predicted to be activated: lipolysis, fatty beta oxidation, cholesterol biosynthesis, glutathione methylation, detoxifications. And we know the hepatocytes perform important roles in the normal liver. And these metabolic pathways are all predicted to be activated in hepatocytes versus others. And they are inhibited in the human liver. So we can ask a question, which is a feature that has been released in QIAGEN IPA at the end of March, of looking at particular pathways of interest that has been predicted to be activated, as I mentioned in the previous slide, and ask the question where that pathway is predicted to be activated when you bring thousands of data sets, right? So from the OmicSoft Land, we have gathered a set of 60,000 comparison expression and put that in our database in IPA. And now we can see where fatty acid beta oxidation is predicted to be activated. This is called an activity plot. All these dots represent a data set. On the x-axis, we have the z-score. On the right, predicted to be activated. On the left, predicted to be inhibited. And this is based on the log p-value. So the higher level, on the most right, the most significantly predicted to be activated. On the left, higher level, the most significantly predicted to be inhibited. Just for the visualization, I look at that point. This is a particular data set. It was about high-fat diet, and this is a treatment where you use a PPAR agonist in specific conditions. So let's explore where fatty acid beta oxidation is mainly predicted to be activated. So we use a tool that will basically try to give us an idea of enrichment of keywords, right? That's what we try to gather. So I have selected on that previous slide a set of data sets that will give me a z-score superior at 2. So we have a very high confidence that the fatty acid oxidation pathways is going to be predicted to be activated. And we are going to look at a set of data set that has been imported from OmicSoft that came from Disease Land and Oncoland. It seems to be a little bit complex right now, but Disease Land and Oncoland are a set of comparison expression in different oncological or many other type of disease. We have gathered them, normalized them, curated them, sent that to IPA, and now we can compare the profile of prediction between our own data set and these data sets. So I have selected filter z-score 2, specifically in Disease Land and Oncoland, and now we are going to look, when we evaluate the metadata, where are the terms that comes the most, that will give us an idea in what type of data set FABO is predicted to be activated. And in fact, when you look at that, FABO activity is significantly enriched with high-fat diet key terms and PPAR agonists. PPAR agonists are anti-diabetics. These PPAR nuclear receptors are indeed involved in the control of metabolism and inflammation in metabolic disease and immunity. So just looking at the enrichment of keywords coming from the metadata that have been processed from OmicLand, we can tell you that fatty acid beta oxidation pathways are predicted to be activated when you have that context of high-fat diet or usage of PPAR agonist. That can give you some clue. In that hepatocytes, we have looked at the top gene markers. And just looking at the 16 genes here, with their expression uniquely in hepatocytes, all of these genes here, you recognize many of them. It encompasses so many functions that the liver is doing, particular metabolism of cholesterol, triglyceride, lipoprotein, fatty acid, or also transport, coagulation, blood vessel formations, fibrosis, inflammatory processes, and many others. So when you have a SERAT, top gene markers, you can extract meaningful information by going back from OmicSoft, scatter color view, and IPA, the gene view, where you will generate a lot of new knowledge. Let's look at particular cell type that we have compared to the rest, cholangiocyte. And when you have the scatter color view, you identify the top gene marker, EpCAM+, which fits exactly what the paper has described. This is the cholangiocyte, a small number of cells very well identified and characterized, we can try to understand what would they do. We know these cells are normally quiescent in the liver. And in fact, they do respond to injury or stress by enhancing their proliferation. Here, this is one of the top canonical pathways that has been predicted by IPA to be inhibited. So we are talking about the pathways called acute phase response signaling. And that pathway is predicted to be inhibited. And in fact, indicating the absence of stress, we are looking at a single cell sequencing from a normal human liver. That pathway should be predicted to be inhibited. This is a strong p-value, a strong z-score. So in a normal situation, the cholangiocyte should not trigger that particular pathway. And that is confirmed by the QIAGEN IPA analysis. Now we would like to understand in very simple terms what will be, between quote, the transcriptional program. What is driving all that particular expression file? And here I'm just giving you a screenshot of a comparison of what we call upstream regulators. To simplify, transcriptional programs that are the drivers. And here, this is a hierarchical clustering of all the different comparisons, hepatic stellate versus others, endothelial cells versus others, and many other situations here, hepatocyte versus others. And we are going to represent here the pattern of activity of that particular gene, VEGF-A, in each of these different clusters. So what you need to remember is the orange color indicates to you a pattern of activations, a blue, a pattern of inhibitions. So VEGF-A is the most predicted to be activated in liver, sinus, endothelial cells here, has the highest z-score and highest p-value. Let's look at what's happening in that liver, sinus, endothelial cells. We know they control the tolerance in normal liver, but they are as well implicated in angiogenesis in normal liver, human liver. And this is what we're going to see using that VEGF-A as a driver. So here, going back to the scatter color view, we identify one of the group of the endothelial cells by the marker CLEG4G+, here. In that particular group, we have identified VEGF-A, as I mentioned in the previous slide, to be predicted to be activated. Let's imagine we put all the genes that are known to be associated with that VEGF-A downstream of it, connected to them, and we can look at what would be the biological process being statistically significant. The top one is angiogenesis. So that network indicates what? VEGF-A is predicted to be activated, it's in orange, based on the pattern of expression of all the genes you see on that network. And that network drives angiogenesis up. That's very interesting. Now let's look at VEGF-A. Where is it expressed exactly? Well, VEGF-A is expressed mainly in the hepatocytes group and almost none in the endothelial cells. That gives you an idea of a possible signaling from one cell to another. And that would not have been discovered if you don't have the two tools together, the QIAGEN OmicSoft Land and the QIAGEN IPA. Let's analyze the Kupffer cells. We know they drive the host defense, the iron metabolism, and phagocytosis, as well as many other functions in the liver. On the left, three scatter color view that are showing the Kupffer cells markers that has been identified in the paper as well, CD5L, Marco, VISIC4. All of these markers are having a role in immune functions. We have looked at the same idea. We have compared all these different clusters and look at a particular upstream regulator, quote the transcriptional program if you wish, And we have identified SPI1 as the most predicted in the Kupffer cells, here in orange. Not at all activated in hepatocyte. It's blue. It's inhibited. That factor is predicted to be activated in the Kupffer cells. And you can tell here, when you look at the scatter color view, where it is expressed, it's actually expressed as well in the Kupffer cells. So that differs from the previous example where one gene is not expressed yet, drives a program on another type of cells. And in that case, one factor expressed in the type of cells and drive the same program within the same cells. That Kupffer cells, we know they are involved in immune responses. And you can see that here. I've done the same, created a network driven by SPI1, predicted to be activated. When we grow a disease, it's a tool we have in QIAGEN IPA, we look at the most significant function, and immune response of white cells are predicted to be activated. So that factor going up boosts the immune response of leukocytes. We can detect that in the Kupffer cells. But we can look also at another type of factor that was interesting to us is PF4. PF4 is a protein likely supplied by the blood and activate targets in the Kupffer cells. PF4 has the highest prediction of activations in the Kupffer cells, much lower in the other group, inhibited in the hepatic cells, and non-existent in the other set of clusters that we see here. PF4 is not expressed in the normal human liver. You can see that absence of a dot in any of the cluster types. Yet, PF4 is predicted to be active in the Kupffer cells. So one question was, if you put the target of PF4, which are represented here by these immune proteins, CXCL3, CXCL8, and many others, you connect them through PF4 because based on the pattern of expression of these genes, we have identified that PF4 is predicted to be up. We have asked the question, let's connect the most significant canonical pathways that we have defined previously. Well, TREM1 is in orange, and hepatic fibrosis signaling is predicted to be activated as well. So we know that PF4 factors, which is predicted to be activated in Kupffer cells, can drive the hepatic fibrosis and launch some part of the innate immunity through TREM1 as well. So you can associate lack of expression, prediction of activation of a particular factor, and a set of canonical pathways via a set of genes connected. You can look also the predicted activity using the Z-score of these two different factors, PF4 and SPA1, when you overlay that on the testing. That's an exercise that we have done here. So you can quickly see that for PF4 hepatocytes, there is not even a Z-score. We don't know. But for some other cells, like the NK cells, it's the gamma delta T cells, it's predicted to be inhibited. And same for SPI1. SPI1 is predicted to be inhibited in hepatocytes, strongly activated in gamma delta T cells, and some other macrophage and Kupffer cells. We can also discover the PS4 expression using the OmicSoft Land Explorer. We have looked at the GTEx, which look at the 51 normal human tissues. PF4 is heavily expressed in the blood, not in the liver. And we can specifically look at the expression in normal immune cells, looking at blueprint B38. Alpha beta T cells are strongly expressing that particular molecule, much less for the other group of cells, including M1, M2 macrophages. All of that is realized by going to the gene view in QIAGEN IPA PF4, clicking on the link to GTEx or to blueprint. It's extremely easy, very simple. We have looked also at PAX5, which is predicted to be activated in the resident B cells in normal human liver. Same as PF4. PAX5 is not expressed at all in a normal human liver. Yet, we can predict a strong pattern of activity in the cluster of B cells versus the others. Stronger Z-score, strongest p-value for PAX5, predicted to be activated in the B cells, absent or inhibited in other type of clusters. When you look at the network driven by PAX5 and you put the genes downstream from which we can predict the pattern of activity of PAX5, and you set all these molecules that are upregulated in the B cells or downregulated in the B cells when you compare to others, and you put them in all the different scatter color view, you realize by looking at red circle or green circle, and this is the B cells cluster, in which these particular molecules are expressed. And effectively, these sets of molecules are expressed in the B cells, and these sets of molecules are downregulated, not expressed in the B cells. So you can go back and forth to a network of interest, a factor of interest in a particular cell type of interest, and visualize that in the scatter color view. Same, we have looked at the expression in GTEx and Blueprint. This particular factor heavily expressed in naive B cells, immature, memory, and class switch B cells, right? Yet, I told you in our case, in the normal human level, we didn't see that much of, if known, PAX5 expressions. Maybe it's due to the fact that these specific B cells are not really present. We may have another type of marker to define which one, but these ones were not probably present as well. It's heavily expressed in the spleen, the blood, and small intestine, using a quick view on GTEx as well. We were interested in another factor that were this time activated in 3 cell types: alpha, beta, gamma, delta, and NK cells, the immune cells per se, that are revealed in that single cell sequencing. And NLC5 actually play a role in cytokine responses and antiviral immunity. And it's predicted to be activated, as you can tell by the orange color, in all these 3 types, and strongly inhibited in the hepatocyte. If you look at the expression of an LRC5 in the scatter color view, you realize effectively the presence of alpha beta T cells here, some macrophages, some endothelial cells a little bit. We have asked the same question as for the pathways. An LRC5 is predicted to be activated, but in what situation? Can looking at 60,000 data sets tell us anything, any insight? And the answer is yes. So here, this is a plot that looks at 7,000 analyses in which an LRC5 is predicted to be activated, this score superior to 2, or predicted to be inhibited, this score inferior to -2. We can look at a specific metadata. Here, this is a interferon alpha versus control in a fibrosarcoma type of experiment. Well, let's explore in our case, where we think that an LRC5 is expressed. We are going just to set this score superior to 2. So we're asking the question, show me the enrichment of keywords in which that particular NLRC5 is predicted to be activated. Same as before, we're going to evaluate the metadata, give us an idea. Well, not surprising when you look at the case treatment, that NLRC5 is predicted to be activated when the treatment is interferon gamma or interferon alpha, and in response to viral infection. That gives you the notion that that compound is pretty important in immune response in general. And to confirm that, you look at the set of analyses in which that compound is predicted to be inhibited. To quickly go about, if you evaluate the metadata with setting certain Z-score, you realize that that compound is inhibited when you have an acute respiratory infection. In that particular case, when you have the chance to have QIAGEN IPA, you will look that type of experiment was including rhinoviruses and coronaviruses, as we know. It's a dramatic situation right now. So that compound is predicted to be inhibited in the case of viral infections. So it can tell us that it's an important player in immune responses. If you put that compound on a network, as well as interferon gamma, which we know right now that they are connected based on the metadata, and we did find a connection in QIAGEN IPA that tie that interferon to an LRC5, and you put that on the networks. All these genes are known to be downstream of interferon or an LRC5. when you put them together, and they are both predicted to be activated, viral infection going down. So it's a very simple way of expressing the fact that interferon gamma and LRC5 are actually connected and participate in the antiviral response in the liver. Now, I told you many times since the beginning of that webinar that we have the possibility of comparing our own data set we have done, let's say hepatocyte versus other, B cell versus other, to a set of 60,000 other curated comparison expression. Here it's called analysis match. And that helps us to discover many hidden biology by looking at other data sets. What's going on in other types? Can we identify key regulator pathways? Can we get greater insight? Can we have confidence in our results? And just doing that by comparing to the rest of the world. So to simplify, thousands of these comparison expression data sets coming from zero TCGA as expressed in my first part of the talk have been processed using the same pipeline, and they have been sent into QIAGEN IPA in a repository. And along with the 7.1 million findings we have, you can tell you can build a very strong biological insight. And we can do that because we can look at different disease, different tissue, different expression, different RNA-seq data sets. So let's go to what we know right now. We compare the gamma delta T cells versus others. And we have selected two sets of land called TCGA, very well known by everybody, and OncoGeo, which encompass a lot of comparison expression in oncological situations. And I have to ask the questions, show me in what my gamma delta T cells, normal situations versus others, can compare to. And it does compare to liver and bile duct cancer liver. It's very strange. But it does compare at the canonical pathways level and the transcriptional level with some important difference. So in other words, I have looked at the gamma delta T cells versus other comparison. And I ask the question, what are the other data sets in TCGA and OncoGeo can compare to a very high level of similarities for the canonical pathways prediction and upstream regulator predictions to my own data set? Well, liver and bile duct cancers compare. As I said, with some differences, and the differences are here. We have three components. One I didn't mention, but just for the argument I show that, NLRC5, interferon gamma, and TBX21. All of them are involved in human and bile duct cancer, apparently. All of these compounds are predicted to be activated in a normal human liver in these gamma delta T cells. But you can tell when you compare these upstream regulator programs, interferon gamma is the only one to be predicted to be activated in these oncological situations, liver cancers, bile duct cancers, and many others. So it's very interesting because when you have the gamma delta T cells, these three compounds in the normal situations without viral insult or other type of injury or other type of disease like cancers, these three components are predicted to be activated. But NLRC5 and TBX21 are not predicted to be activated. They are not even present, apparently, in these liver cancer and bile duct cancers. So that tells us, as an hypothesis that needs to be validated, to fight this type of disease, for instance, you will need the three components to be predicted to be activated to make it work. When you have only one, well, cancer is still there. We can look also in a gland called human disease. What is the expression level in particular disease of the liver? These compounds are expressed: TBX21, interferon, and NLRC5. And here, this is just a very quick representation. Maybe you don't see enough, but in situation of inflammatory disease, TBX21 is highly expressed, interferon much less, and NLRC5 a lot. So going from that comparison of upstream regulator from a normal set cell type versus other in disease situations. You can also look at the expression of this particular compound in a setting that are not relevant to our case here, but inflammatory disease or other type of injury. So to conclude here, I hope I could explain that we can identify biological clusters easily in our single-cell Land project. We have identified many upstream regulator and biological processes that are involved in a cell-specific manner. We have identified as well biological cluster that could be connected to important expression data in other data sets, such as cancer and other diseases, to highlight some kind of correlations. And we have also discovered hidden biology by understanding the behavior of molecules, pathways, and disease and function across a set of thousands of other comparisons. So on that note, thank you very much all. I hope that webinar will be well appreciated. And if you have any questions, we have these contact and these support and key phone number. Thank you very much.